What we'll do here is take a look at cellular metabolism. You've probably already taken a look at cellular metabolism of mostly animal and plant cells in a previous class. So what we're going to do here is focus on cellular metabolism that occur in bacteria. It turns out that there's some bacteria out there that do really similar things as animal and plant cells when it comes to their metabolism. But if you consider bacteria as a whole, they can do all kinds of unique, really cool metabolic things that animal and plant cells could only wish they could do. So we'll focus here on some of the unique things that bacteria can do when it comes to metabolism. And before we do that, it might help to kind of throw down a definition for cellular metabolism so that we know what we're talking about. One way of defining cellular metabolism is that it's all of the chemical reactions that are happening inside of a cell that are needed in order for that cell to stay alive. In order to do these chemical reactions, you need substrates, right? You need molecules that participate in the chemical reaction. So maybe another way of thinking of metabolism, it's the breaking down and building of molecules that are required in order for a cell to stay alive. And so if you think about it, cells really need just a few types of molecules in order to stay alive, and we'll list those here. One type of molecule that a cell needs access to in order to stay alive is a molecule that contains high energy electrons. And so this is the symbol that we'll use for energy from here on out. So whenever you see a capital E, that means energy. To kind of go along with that, another type of molecule that cells need access to in order to stay alive is a molecule that has a pull for electrons. And so you might be wondering, well, how come cells need electrons in order to stay alive? Well, if you remember, the molecule that most cells use in order to power all of their activities is a molecule called ATP. And most cells, in order to make most of the ATP, that's done through the movement of electrons. So here's what we mean by that. Again, in order to make ATP, most cells, they take an electron from some molecule that has high energy electrons, they break it down so that that electron can move towards some molecule that has a pull for that electron. Whenever that electron moves, or anything that moves, represents kinetic energy, and it turns out that cells can kind of harness that kinetic energy and use it to make something else that has a lot of energy, which is ATP. So again, the importance of having access to molecules that make electrons move is that it's the movement of the electron that basically generates the ATP, most of the ATP inside of cells. If you take a look at the leftovers here, so this molecule here, after it got broken down, the leftovers can either be further metabolized or they are just released as waste products. This molecule here, after it's accepted the electron, it's already done its job, right? The ATP has already been produced, so this no longer has a function, so it's also usually released as a waste product. So here's one of the sources of metabolic waste products inside of cells. So again, kind of going back to our list of what types of molecules do cells really need in order to stay alive. They need these two types of molecules here, something that gives up electrons and something that pulls electrons towards it. The reason why is because that's how most cells make most of their ATP. And so because molecules that have high energy electrons are responsible, take part in producing ATP, another term for an electron source is an energy source. So these two things are used kind of interchangeably. So if a molecule is serving as a cell's electron source, that's the same thing as saying that that molecule is serving as the cell's energy source. Another type of molecule that a cell needs access to in order to stay alive is a molecule that contains carbons. So you might be wondering, well, why is that so important that a cell have access to a molecule that contains carbons? Well, if you take a look at the major structural components of any type of cell, of bacterial cells, animal cells, plant cells, here's what you'll see. All right, so here are some examples of the major structural components that you'll find inside of all the cells. All cells have some sort of nucleic acid, and nucleic acids are comprised of nitrogenous bases that are all linked together. And if you take a look at the atoms that make up nitrogenous bases, check it out. One of the major atoms that you find inside there are carbon atoms, a whole bunch of C's. Same thing for lipids. All cells have lipids inside of them, and you see that lipids have a whole bunch of carbons in them. Amino acids, which are strung together to make proteins, you see that amino acids also have carbons in them. So we're saying here that in order to stay alive, a cell needs access to carbon atoms. And the reason why it needs access to carbon atoms is because carbon atoms are basically used to build everything that you find inside of a cell. So that's why this is important. Those molecules containing carbon atoms serve as building blocks of a cell. And then one more thing that we'll tack onto the list. Basically, there's miscellaneous molecules that a cell needs access to in order to stay alive, and we'll just kind of group them all together here as our number 4 on the list. So here are some other things that cells need access to in order to stay alive. They need nitrogen, sulfur, and phosphorus, because those are atoms that are also used to construct most everything that you find inside of a cell. Iron and magnesium are used to help a lot of the enzymes that run a lot of the reactions that occur inside of cells. All right, so these things are needed because they serve as building blocks, and S and P do, or cofactors for enzymes. Iron and magnesium are the cofactors for enzymes. So then these are all of the molecules that all cells basically need access to in order to stay alive. There's all kinds of different chemical reactions that occur inside of a cell in order to process all of these molecules. And so maybe one way of making sense then of all of the chemical reactions, out of all of the metabolic reactions that are happening inside of a cell, is to, if you could somehow categorize those chemical reactions, that might make it easier to kind of make sense of all of the different types that are happening inside of a cell. So what we'll do next here is show you some different ways of categorizing all of these metabolic reactions. One way of categorizing metabolic reactions is based on generally what's occurring. So there's some metabolic reactions that involve breaking stuff down, and there's some that involve building stuff up. So metabolic reactions that involve the breaking down of something are referred to as catabolic reactions. On the other hand, metabolic reactions that involve building something up are called anabolic reactions. So the reason why a cell might want to catabolize something or break it down is so that, for example, electrons can be released from it so that it can be used to make ATP. Another reason why a cell might want to catabolize something or break it down is if it wants to release carbon atoms from it so that it can build other cell parts. A reason why a cell would want to anabolize something, to build something up, is so, for example, it could take some of the broken down carbon atoms and build them together, put them together to build bigger things. So these two types of metabolic reactions, they're different, but one is needed in order for the other one to occur, right? So catabolic reactions are needed to make ATP, energy, that's needed in order to power the building of something. One of the ways that I kind of keep these two straight in my mind is I always think of anabolic steroids. So whenever you think of someone that's using anabolic steroids, they're really pumped up, really ripped. And so the reason why they're ripped is because they have built up muscle. So anabolic steroids has the term anabolic in it. Anabolic means to build something up. Catabolic is then the opposite, to break something down. So this is one way of categorizing then metabolic reactions that are happening inside of a cell. Another way of categorizing all of these reactions that are happening inside of a cell is according to what type of molecules are used in each metabolic reaction. So all of the molecules that you see listed here, as far as ones that are really needed in order for a cell to live, they can be categorized as one of two types of molecules. There are some molecules that are organic, and by definition, if a molecule is organic, that means that it contains both carbon atoms and hydrogen atoms. On the other hand, though, there are molecules that are considered to be inorganic. So if they're inorganic, they lack carbon or hydrogen or both. So just to kind of drive home the point, what's the difference between organic and inorganic? Well, here are some examples of organic molecules. Nitrogenous bases, which are linked together to make nucleic acids, these are considered organic because they fit our definition, right? They have both carbon atoms and hydrogen atoms. Lipids are also organic because they have both carbon atoms and hydrogen atoms. Amino acids, which are used to make proteins, are organic because they have carbons and hydrogens. And carbohydrates are organic. You can tell from the name that it has both carbons and hydrogens, and you can also tell from the molecular structure. There are carbons and hydrogens. One molecule that people kind of have a tough time categorizing is CO2, carbon dioxide. Turns out that CO2 is not organic. It's inorganic because even though it has a carbon atom, it does not have hydrogen. So that doesn't fit the definition of organic. Right, so again, what we're doing here is we're kind of trying to categorize things so that we can make it kind of easier to understand the different types of metabolic reactions that happen inside of a cell. This is one way of categorizing those things. Another way you can categorize stuff metabolically speaking is you can categorize each type of cell based off of what types of molecules they can metabolize. So here's what we mean by that. So we're saying here that one of the types of molecules that a cell needs access to in order to stay alive is a molecule containing high energy electrons. And again, the reason why a cell needs access to a molecule like this is because when it breaks down that molecule to release the electron, it's the movement of the electron to another molecule that generates energy that's needed to generate ATP. So different types of cells use different types of molecules as their electron source. There are some cells that use organic molecules like carbohydrates, proteins, and lipids as their electron source. And so in order to identify these types of cells, they're given a name. And so what these types of cells are called, they're called chemoorganotrophs. Right? So by definition, a chemoorganotroph is a cell that uses organic molecules as its electron source. There's other cells that use inorganic molecules as their electron source. And here are some examples of inorganic molecules that are commonly used by these types of cells as electron sources. By definition, if you're talking about a cell that uses an inorganic molecule as its electron source, it's called a chemolithotroph. So if you break the term down, it kind of makes sense. Chemo tells you that these cells use some sort of chemical compound as their electron source. Litho refers to rock, and rocks are inorganic. And then troph means to feed on something. And so these guys are feeding on these types of molecules and using it as their electron source. One other category as far as electron sources are concerned are this. There are some cells that use various low-energy organic and inorganic molecules as their electron source. So you might be wondering, well, how can a cell use something that has low energy in order to squeeze ATP out of it, basically? Well, what these cells do is something kind of cool. They can take something that kind of intrinsically has low energy and make it have higher energy by using the energy from the sun. So these cells can absorb light energy in order to energize normally low energy molecules to make them have high energy electrons. And so whenever you're talking about a cell like this that takes normally low energy molecules and energizes them using light energy, they are called phototrophs. And so photo means light, so it kind of makes sense why these would be called phototrophs. You can also categorize cells not only based off of what they use as an electron source, but also what they use as an electron acceptor. And so here are some of the categories for that. There are some cells that, as their electron acceptor, they use inorganic molecules like oxygen, nitrate, sulfate. And so by definition, if you have a cell that uses an inorganic molecule as its electron acceptor, it's called a respirator. In other words, it's said to be performing respiration. It turns out that there's two types of respiration. If the inorganic molecule that's being used by this type of cell happens to specifically be oxygen, then that type of respiration is called aerobic respiration. If, on the other hand, the inorganic molecule that's used is something besides oxygen, then it's called an anaerobic respirator. So anaerobic respirators would use things like nitrates and sulfates, so something besides oxygen gas as its final electron acceptor. Another category when it comes to electron acceptors that can be used by cells is this. There are some cells that, instead of using inorganic molecules, use something organic as its final electron acceptor. And an example of an organic molecule that can do that is pyruvate. And so by definition, a cell that uses an organic molecule as its electron acceptor is called a fermentator. In other words, it's said to be undergoing fermentation. And so again, what we're showing you here is that you can categorize cells based off of what types of molecules they use as an electron source and as an electron acceptor. If you want to get really fancy, you can kind of combine these terms to kind of give you an overall picture of what a cell does metabolically speaking. So here's an example of how you can combine some of the terms that we just took a look at. For example, you could say that there are cells that are chemoorganotrophs that perform aerobic respiration. And so if you consider each one of these terms, it'll tell you exactly what it's doing as far as its metabolism is concerned. Remember, chemoorganotroph tells you that as its electron source, it uses something organic. Respiration tells you that it uses something inorganic as its electron acceptor. This aerobic here tells you that specifically the inorganic molecule that it's using as its electron acceptor is oxygen. And by the way, this happens to describe what animals do, what we do. We are chemoorganotrophs that perform aerobic respiration. Sometimes our muscle cells do fermentation when it needs a little bit extra boost of energy, but for the most part, this is what we do. And some bacteria can do this as well. All right, we're still trying to categorize things. So that's just one example of how cells can be categorized. Turns out that cells can also be categorized according to what types of molecules they use as carbon sources. And so here's what we mean by that. There are some cells that the type of molecule that they use as their source of carbon are inorganic molecules, specifically CO2. And so when you're talking about a cell that uses CO2 as its source of carbon, it's called an autotroph. So that would make sense. So these types of cells can, well, first of all, autotroph, if you break down that term, auto means self, and then troph means to feed. So these are cell feeders. It would make sense that these types of cells are called cell feeders because they can just basically sit there and absorb the CO2 from the air and use that as their source of carbon. They're feeding themselves carbon by just sucking in the CO2 from the air. The other category, as far as describing carbon sources are concerned, is this. So there's some cells that don't use inorganic molecules. Instead, they use organic molecules like carbons and proteins and lipids as their source of carbon. And so whenever you have that type of cell is called a heterotroph. And so again, you can categorize cells based off of what types of molecules they're using for all of these different things. If you want to be really, really fancy, you can combine these three, you can combine terms that describe these three types of molecules in order to really describe what a cell is doing metabolically. So here's an example of what you could do. You could say that a cell is a chemoorganoheterotroph that performs aerobic respiration. So you probably just almost ran out of breath just listening to that, but if you just break down this almost paragraph, it'll tell you exactly what the cell is doing. So if you consider this term chemoorganotroph, remember that means that this cell is using an organic molecule as its source of electrons. Respiration again means that this cell is using something inorganic as its electron acceptor. Aerobic tells you that specifically the inorganic thing it's using as its electron acceptor is oxygen. And we've just learned that this term here, heterotroph, tells you that this cell, for its source of carbons, it uses a molecule that is organic. Right? So this really long kind of description here is really useful because it tells you exactly what type of metabolism this cell is doing. And again, it turns out that animals, including us, happen to do this type of metabolism. We are chemoorganoheterotrophs that perform aerobic respiration. Again, sometimes our muscle cells do fermentation, but for the most part, this is the type of metabolism that we can do. We can't do chemolithotrophy or autotrophy. That's kind of the sole realm of bacteria. So bacteria can do all kinds of cool things again that we can't do. So basically what we've just done here is that we've broken down metabolism into different categories so that hopefully that'll make it more easily, or make it easier to manage the understanding of the different metabolic processes that different cells can do. What we'll do next is take a look at each one of those categories and see what all a cell has to do in order to do that type of metabolism. So the first category that we'll take a look at here is this. We'll take a look first at cells that can metabolize organic molecules and use oxygen. And so to relate this to the terms we just explained, you know that if a cell is able to metabolize organic molecules, that means that it could be one of three things, or all three. So if it's metabolizing organic molecules, that means that it could be a chemoorganotroph, could be a heterotroph, or it could be a fermentator, depending on what it's using the organic molecule for. Right? So again, remember that if it's a chemoorganotroph, that tells you that it's using organic molecules as its electron source. Heterotroph tells you that it's using organic molecules as its carbon source. Fermentator tells you that it's using organic molecules as its final electron acceptor. If a cell is metabolizing oxygen, oxygen is used as a final electron acceptor because it's very electronegative. So if it's using oxygen, that tells you that it's an aerobic respirator. So again, we're going to take a look at next at cells that do this type of metabolism. So kind of the gold standard molecule out there as far as organic molecules are concerned is glucose. So what we'll do is use glucose as an example of how organic molecules are metabolized by cells. And so if you remember the structure of glucose, it has 6 carbons in it, and then some other functional groups associated with it. So here's glucose in its linear form. Remember that glucose in solution also forms a ring structure that looks like this. The thing to point out here is that organic molecules like glucose can be valuable to a cell for basically 2 different reasons. Right? One reason why a molecule like glucose is really valuable to cells is because it has carbons in it. And remember, the carbons are really important because they're used to build basically anything that you find inside of a cell. So one reason why glucose is really valuable is because it has carbons. Another reason why a molecule like glucose is really valuable is because it has a lot of these types of bonds. It has a lot of carbon-to-carbon covalent bonds, and it has a lot of carbon-to-hydrogen covalent bonds. And so you might remember from cell biology that these types of covalent bonds, those between 2 carbons, those between a hydrogen and a carbon, those are high energy bonds. In other words, if you were able to release the electrons that are involved in that bond, those electrons have a lot of potential energy that can be used by that cell to do other things. So another reason why glucose is so valuable for a cell is because it has high energy electrons in it. And so then in order to get those items of value out of that glucose molecule first has to get broken apart. And so basically here's what that involves. Again, glucose, we'll draw it in a really simple form so that we're just showing its carbon skeleton. Remember, it has 6 carbons in it. In order to get everything of value out of that glucose molecule, it has to get broken apart so that things like the high energy electrons in that glucose molecule can be released. Remember that that high energy electron is valuable because if it's an aerobic respirator, at least, what happens is that oxygen that makes its way inside of the cell pulls the electron towards it in order to generate ATP. The thing that actually does the ATP generating, remember, is a series of molecules called an electron transport chain. So it's when the electron is pulled through the electron transport chain by that oxygen. That electron transport chain uses then the energy of the moving electron in order to make a whole bunch of ATP molecules. If you take a look at the leftovers here, so this glucose is getting broken apart. Another reason why a cell would want to break down glucose is so that it can get electrons out of it, and also to maybe break it down a little bit more so that you get these carbon building blocks that can be used to build the major structures inside of a cell. So again, what we're saying here is that glucose is valuable for 2 different reasons. And the reasons are this: breaking it down releases high energy electrons that are used for making ATP. Breaking down glucose also releases or generates these smaller carbon-containing molecules that can be used for building blocks in order to build the bigger structures inside of cells. So then what we'll take a look at next is some of the pathways that a cell might use in order to break down a glucose molecule. One pathway you should be pretty familiar with, because you should have talked about it in cell biology, and that's glycolysis. So if you break down this term glycolysis, literally it means sugar breaking. So this is one option that cells have to break down glucose, and basically all cells on this planet can do glycolysis. And basically what glycolysis involves is this. Basically it breaks glucose down in half. If you remember, you can divide glycolysis into two stages. Right? The first stage of glycolysis is when that 6-carbon molecule is broken in half. In the process, a phosphate group is attached to each one of the halves. During the second part of glycolysis, it turns out that another phosphate group is attached to each one of these molecules, but then those phosphate groups are removed to produce 2 molecules that each contain 3 carbons in them. And if you remember, those molecules are called pyruvates, or pyruvic acids. It's during this kind of second part of glycolysis that the high energy electrons in the glucose molecule become released. So this is the electron that goes towards the oxygen, if we're talking about an aerobic respirator. Remember though that the electron doesn't just by itself go to the oxygen. Instead, there's a molecule that delivers the electron to the oxygen. The name of that molecule is called NAD+. So it's here that NAD+ swoops in basically and picks up that released electron. When it does so, NAD+ not only takes on an electron but a proton to become NADH. So it's that hydrogen atom that now contains that released high energy electron. And so this is the molecule that ends up delivering that electron to the electron transport chain and the oxygen that's awaiting it at the end. If you remember, this second part of glycolysis also releases a little bit of ATP, so that's valuable too. So again, what we're doing here is describing one of the pathways that a cell might use to break down an organic molecule like glucose. It turns out that these leftover organic molecules, pyruvate, can be further broken down. So remember that we're just showing you the basic carbon skeleton here. Pyruvate also has hydrogens in it, so that's why it's considered to be an organic molecule. Remember that there's something called the citric acid cycle, or the TCA cycle, tricarboxylic acid cycle, or the Krebs cycle. So those are 3 names for the same thing. So there's something, we'll use the citric acid cycle. So there's something called the citric acid cycle that can basically squeeze even more carbons and even more high energy electrons out of this glycolysis leftover. In order for pyruvate to enter the citric acid cycle, it has to be modified. So the modification involves getting rid of one of the carbon atoms in the form of CO2, and it involves the attacking on of something called coenzyme A. So after a carbon is removed and after this guy is tacked on, you get something called acetyl-CoA. And so this is the form of pyruvate, basically, that is able to enter the citric acid cycle. And like we mentioned, during the citric acid cycle, even more goodness is extracted from this molecule. So high energy electrons can be extracted from that acetyl-CoA. Those high energy electrons are picked up by a molecule of NAD+ to become NADH. Turns out that there is another electron carrier that cells can use to deliver electrons from one place in the cell to the electron transport chain. That other electron carrier, if you remember, is called FAD+. So some of the high energy electrons that are released during the citric acid cycle are picked up by FAD+ to become FADH2. Again, the red H is where the high energy electron is located. Also, if you remember, the citric acid cycle produces a little bit of ATP, so that's also valuable. And as one of the waste products, the citric acid cycle produces CO2, and we'll come back to this actually a little bit later. So the citric acid cycle, again, is another pathway that breaks down organic molecules to release some really valuable things. And again, what the valuable things are this. So all along the way here, we're seeing being generated some smaller carbon-containing molecules that can be used to build bigger structures inside of a cell. Also being generated here are molecules like these guys, NADH and FADH2, and again, they're valuable because these are the guys that contain now the high energy electron that will be used to make ATP when it's delivered to the electron transport chain. So all kinds of really valuable things are being generated here. It turns out that glycolysis and the citric acid cycle aren't the only ways of breaking down organic molecules inside of a cell. So there's another pathway that you may or may not have taken a look at in cell biology called the pentose phosphate pathway, or PPP. And what the pentose phosphate pathway does is that it breaks down glucose, but in a little bit different way and for a little bit different reason. So if glucose is broken down during the pentose phosphate pathway, one of the things that's generated is a molecule that contains 5 carbons in it. And the reason why a 5-carbon molecule would be really valuable is because it's used to build ribose sugars. Ribose sugars have 5 carbons in it. And you might remember that ribose sugars are really valuable because that's the sugar that you find in nucleic acids. Right? So basically this guy here, this 5-carbon sugar, is used to make nucleic acids, which are used to make DNA and RNA. Some of the other things that are generated by the pentose phosphate pathway include this. So you might be wondering, well, where did that 1 carbon go? Glucose has 6 carbons. This guy now only has 5. Where did the 6th one go? It turns out that it's released during the pentose phosphate pathway as a molecule of CO2. So that's where that carbon atom went. Another thing that's generated during the pentose phosphate pathway is a molecule called NADPH. And the reason why NADPH is valuable is because this molecule is required by anabolic reactions. So this molecule basically helps power anabolic reactions, which again are reactions that are involved building up things. So this guy is valuable for reasons we just explained. This molecule here is pretty valuable. CO2 is actually just released as a waste product. And actually, coming back to this molecule here, this molecule is valuable for another reason too. So depending on what the cell needs at the time, it can do a couple of different things with this molecule. It can either use it in order to make nucleotides, or it could break down this 5-carbon molecule to produce a 3-carbon molecule and a 2-carbon molecule. And this 3-carbon molecule can be used to construct this molecule over here that is required for glycolysis to happen. Right, so this again, this molecule here is one of the end products of the pentose phosphate pathway. Depending on what the cell needs at the time, it can do a couple of different things with it. So again, what we're showing you here are we're showing you different pathways that cells can use in order to break down organic molecules like glycolysis. How about if we sum up then the 3 pathways that we're taking a look at here? So we mentioned that glycolysis is a way of breaking down organic molecules, and what glycolysis does when it breaks down an organic molecule is that it makes things like NADH, it makes a little bit of ATP, and it also makes smaller carbon-containing molecules that are used as building blocks to build bigger things like amino acids and lipids. We mentioned that there's this thing called the pentose phosphate pathway that could also be used to break down glucose. And what the pentose phosphate pathway does is that it produces molecules like NADPH. We mentioned that's required for anabolic reactions. And it also produces smaller carbon-containing molecules that are used as building blocks to make things like sugars and amino acids. So if you compare just glycolysis to the pentose phosphate pathway, again, these are 2 different ways of breaking down glucose. You might be wondering, well, why would a cell need 2 different ways of breaking down glucose? Well, what's the difference between the 2? Well, if you take a look at what's generated during glycolysis, it makes things like NADH, which has a high energy electron in it that can be used to make ATP. And also some ATP is directly made by glycolysis as well. So if a cell is in big need of energy, if it's in big need of ATP, then it would use glycolysis probably to break down that glucose, because glycolysis makes just that, it makes a lot of energy. If on the other hand a cell is more in need of building blocks. If it's in need of carbon molecules in order to build amino acids and lipids, then it would probably break down a glucose molecule by using the pentose phosphate pathway. Because if you take a look at what the pentose phosphate pathway produces, it doesn't really make high energy things like ATP and NADH. Instead, it makes building block molecules that are used to make the stuff that we just talked about, right? Amino acids and lipids and nitrogenous bases. So these are two different ways of breaking down glucose. Depending on what a cell really needs at the time, it'll either use glycolysis or the pentose phosphate pathway. The other pathway that is on this screen is the citric acid cycle. And so to sum up the citric acid cycle, what it does is that it takes the glycolysis leftovers, namely pyruvate, and uses it to make NADH, FADH2, a little bit of ATP, and also some small carbon-containing molecules that can be used as building blocks. So cells that metabolize organic molecules use these pathways in order to break down that organic molecule. The example of an organic molecule that we're looking at here is glucose. What we'll do next is kind of remind you, though, that glucose isn't the only organic molecule that cells can metabolize, if it's a cell that can metabolize organic things. So there are other organic molecules that can also be used to provide carbon building blocks and energy. There are things like cellulose, starch, and disaccharides like lactose. So basically what cellulose and starch are, if you remember, they're the storage forms of energy. So basically cellulose and starch, they're both made up of a bunch of glucoses all strung together. So cells can then also use these, and of course lactose is a disaccharide, so it's two sugars strung together, glucose and galactose. So what we're saying here is that if a cell can metabolize organic things, it could also use things like this. In order to use things like cellulose and starch, though, each one of these glucoses has to be kind of broken off of that bigger structure so that it can enter glycolysis. And there are enzymes that are responsible for doing that. So in order to break down cellulose to release the individual glucose molecules, an enzyme called cellulase is required to make that glucose. In order to break down starch to produce individual glucose units, an enzyme called amylase is required. And also there are enzymes that break down disaccharides to release the individual sugar units. So it turns out that not all cells have these enzymes, cellulase and amylase. So depending on the type of cell, it would be able to metabolize some, none, or all of these different forms of glucose. Another type of organic molecule that can be metabolized by cells are lipids. So lipids could also provide energy and carbon building blocks. So here we're taking a look at a type of lipid called a triglyceride, or a fat. And so if a cell is going to metabolize something like this in its metabolic processes, basically what has to happen first is that this fat has to get broken apart. And the way that it's broken apart, it's like this. It's broken apart so that the glycerol head is removed, and then the fatty acid tails are removed. It turns out that this glycerol head that contains 3 carbons in it can be used to construct this molecule here, 3-phosphoglycerate. And basically that's how lipids are used, or that's how lipids are broken down, right? It can be broken down to release this glycerol molecule, which is used to make this. This molecule then can enter glycolysis basically halfway through here in order to make a whole bunch of NADHs, and then NADHs over here, and some ATPs in the citric acid cycle as well. The fatty acid portion of the lipid molecule can be broken down even further to produce acetyl-CoA, and so that's how this portion of a lipid then ends up getting metabolized. It enters at this point in this pathway that we're taking a look at. Not only can sugars, carbohydrates in general, not only can carbohydrates and lipids be metabolized by cells that metabolize organic things, proteins can also be metabolized. And basically the way proteins are metabolized, they are first broken down into their smaller parts, which are amino acids. And the amino acids then can enter this pathway somewhere basically in the middle. And one place where amino acids can enter is in glycolysis. So it can enter after about the 3rd or 4th step of glycolysis in order to produce all of these really valuable things that can be produced from organic molecules. So the take-home point of all of this can be summed up by this little paragraph here. Polysaccharides, disaccharides, lipids, and proteins, those can also be broken down into smaller parts that then enter midway through glycolysis or the citric acid cycle. So again, the point here is that it's just not glucose that can be used by these cells that metabolize organic things. All of these other organic molecules can also be metabolized. So next, what if we just kind of reset by returning to this more streamlined picture showing 3 different pathways that are used in order to break down an organic molecule like glycolysis, or like a glucose. Remember again that one of the reasons why a cell might be interested in breaking down something like glucose is so that it can generate these smaller carbon-containing molecules that can be used as building blocks to make other bigger molecules inside of a cell. It turns out that each one of these pathways, glycolysis, the citric acid cycle, and the pentose phosphate pathway, they all produce intermediates that can all be used to make other bigger things inside of a cell. So here's what we mean by that. If you consider just glycolysis, right, so the idea here is that carbon-containing molecules are made that can be used as building blocks. All of these pathways make these carbon-containing molecules. If you consider just glycolysis, right now what you're taking a look at is a really simplified version of what occurs during glycolysis, but if you take a look at a more detailed version, here it is. Glycolysis starts with a glucose molecule that goes through a whole bunch of stuff before finally at the end 2 pyruvates are produced. Glycolysis produces all kinds of intermediates that can be used as building blocks to make other bigger things. And so by intermediates, what we mean are these different molecules that are produced as glucose basically gets processed during glycolysis. All of these things can be used as building blocks to make things like amino acids and lipids. Same thing for the citric acid cycle. If you take a look at the actual steps of the citric acid cycle, or sorry, how about if we take a look at the pentose phosphate pathway first? So same thing with the pentose phosphate pathway. If you take a look at the actual individual steps that occur during this pathway, you'll see that there's a whole bunch of molecules that end up being produced. All of these carbon-containing molecules can also be used to build bigger things inside of a cell. And then also the same thing for the citric acid cycle. If you were to actually take a look at each one of the individual steps of the citric acid cycle, you see that all of these intermediate molecules end up being produced. Each one of those could be, or at least a lot of them, can be used to make things like amino acids and fatty acids. So what we'll do here is this. So again, the take-home point is that all kinds of intermediates are produced during these 3 pathways that can be used to build bigger things inside of a cell. What we'll do is just pick some of these molecules and show how they can be used to make some of the bigger things like amino acids and lipids. So first, if you take a look at glycolysis, it turns out that this 3-carbon molecule here that has a phosphate attached to it, this thing that's produced during glycolysis, it's called 3-phosphoglycerate. And it can be used by cells to make amino acids, certain types of amino acids. And remember that amino acids are important because they are used to build proteins. Proteins are important because they do all kinds of things for a cell. For example, they serve as enzymes for a cell. Turns out that this molecule down here, acetyl-CoA, that's also something that can be used as a building block. Specifically, what acetyl-CoA can be used for is to make fatty acids. And remember that fatty acids are important because they're used to make lipids. And lipids are important for cells because, well, for example, there's a particular type of lipid called a phospholipid that is the major component of a cell membrane. So you need these things in order to build membranes, basically. It turns out that there's an intermediate that's produced during the citric acid cycle that can be used to make other types of amino acids. And again, amino acids are important because they're used to make proteins. And then the last thing that we'll kind of highlight here, the last molecule that can be used as a building block is this 5-carbon molecule that's produced during the pentose phosphate pathway. We've actually already mentioned that this guy can be used to build riboses, and riboses are the 5 sugar molecules that are used to make nucleotides. Nucleotides are important because those are the things that are strung together in order to make nucleic acids like DNA and RNA. So again, we're taking a look at cells that can break down, that can metabolize organic molecules. One of the reasons why organic molecules are valuable is because they can produce carbon-containing compounds that can be used to build all of these things here that we just showed you in green. There is another reason why a cell might be interested in breaking down an organic molecule, and that other reason, again, is to release high-energy electrons that are used to make ATP. And so in this picture here, the high-energy electrons have already been produced and picked up by these molecules, NADH, FADH2, and then another NADH during the citric acid cycle. And again, basically what these molecules do is that they deliver that high-energy electron that's been released by these different processes, and it delivers that electron to the electron transport chain. And if it's an aerobic respirator, there's oxygen at the end of the electron transport chain that basically pulls the electron through that chain. So what we'll take a look at next then is the electron transport chain, and basically how it can differ among bacterial cells. You've probably already looked at the basics of the electron transport chain in cell biology, so how about if we kind of start there? You took a look at how the electron transport chain works in the mitochondria of animal cells and plant cells. And so basically this is what the electron transport chain looks like in the mitochondria of animal and plant cells. So to kind of break down this picture, here is the NADH molecule. Remember that this is the guy that picked up the high energy electron that was released during glycolysis and the citric acid cycle. Since we're taking a look at aerobic respirators at this point, you know that it uses oxygen as the final electron acceptor, and so here is where the oxygen rests, right? Aerobic respirators take in oxygen and they kind of position it at the end of the electron transport chain so that it can suck the electrons through. Kind of the path that the electron takes through the electron transport chain is this. So if it's NADH that's bringing the electron to the electron transport chain, where it brings it to specifically is this first complex, complex 1. From there it goes to a molecule called coenzyme Q, which then brings the electron to this complex 3, which then is picked up by this molecule called cytochrome C, which takes the electron to the complex 4. And after it gets there, the electron then is picked up by the molecule that serves as the very last electron acceptor. In this case, it's oxygen. And so if you remember, the movement of the electron through all of these complexes in the electron transport chain, that kinetic energy that's represented by the moving electron is used by the cell to pump protons from one side of this membrane to the other. So these complexes are actually responsible for that. Some of them can take the energy of the moving electron to pump protons from one side of the mitochondrial membrane to the other side. So that's what these H pluses with the arrows on them represent, protons being pumped from here to here. You might be wondering, well, what about complex 2? What is it doing? So you might remember that what complex 2 does is that it's kind of the receiving point if it turns out that FADH2 is the molecule that's being used as the electron deliverer. So if it's FADH2 that's bringing electrons to the electron transport chain, the specific spot in the chain where it brings it to is complex 2. And after FADH2 brings its electrons here, it becomes FAD plus again. After FADH2 has delivered its electron to complex 2, then from there it goes to coenzyme Q, and then it kind of follows the same path as the electron that was delivered by NADH. You might be wondering well, this is all well and good, but where is the ATP made, right? That's kind of the whole purpose of making electrons move through the electron transport chain, is to produce ATP. You might remember that ATP is actually produced by this molecule called ATP synthase, which is also found in the membrane of mitochondria if you're talking about plant and animal cells. Remember that this is the enzyme that actually attaches a phosphate group to an ADP in order to make ATP. This enzyme, though, requires energy to attach these two things together. Where that energy comes from is the diffusion of these protons. So remember, these protons that end up getting pumped from one side of the membrane to another, if you get a whole bunch of them that end up getting built up on this side of the membrane, they want to diffuse away from one another. And one of the places where they can diffuse is through ATP synthase to the other side of the membrane. So ATP synthase just basically accommodates the kind of the need of these protons once they're kind of concentrated in one area to diffuse away from one another. Right? So it's in the ATP synthase that the energy that's represented by the proton gradient gets turned into ATP. So this is the electron transport chain. Right? If a cell is using oxygen as its final electron acceptor and an organic molecule as its electron source, that electron ends up being delivered here by NADH. This is how the ATP ends up being produced. We're taking a look at this electron transport chain in the mitochondria of animals first, because for one, hopefully you're familiar with it, and two, it turns out that there are bacteria that basically perform the same type of electron transport chain. Right? So an example of a type of bacteria that basically does the same type of electron transport chain to make ATP is a species called Pseudomonas, and we'll be working with Pseudomonas quite a bit actually in the lab. One difference though to point out is this. Remember Pseudomonas, it's a type of bacteria. Bacteria are prokaryotes. Prokaryotes by definition do not have major organelles. So Pseudomonas species, when they do an electron transport chain, it's not in the membrane of mitochondria, because bacteria don't have mitochondria. So instead, where bacteria do the electron transport chain is in the cytoplasmic membrane itself. So to kind of give you a visual, again, you learned about electron transport chain in animals in the past, hopefully. And what we're saying is that in animal cells, the electron transport chain happens in this organelle here called the mitochondria, specifically in the inner membrane of that mitochondrion. Bacteria that do electron transport chain, they are prokaryotes, so they don't have any mitochondria inside of them. Instead, where they do their electron transport chain is directly in their cytoplasmic membrane. Right? So, and actually, if you remember, there's this thing called endosymbiotic theory that says that these mitochondria in plant and animal cells are actually bacteria that at one point just made their way inside of this bigger cell. Right? So the electron transport chain happens in the same spot in mitochondria as it does in bacterial cells, because mitochondria basically are bacterial cells. All right, so it turns out that there are other types of electron transport chains that can be performed by other types of bacteria. So for example, E. coli, or how about if we mention this actually before we look at E. coli, you see this very last complex 4 in the electron transport chain. Another term that's used for this particular molecule is called cytochrome oxidase. So complex 4 and cytochrome oxidase, it's the same thing. And the reason why complex 4 is called cytochrome oxidase is because this molecule oxidizes cytochrome C. In other words, this molecule here takes an electron away from cytochrome C. That's why it's appropriate to call it cytochrome oxidase. And we're mentioning it here because one of the ways of identifying Pseudomonas species is by seeing whether a bacterium has cytochrome oxidase. If it doesn't, then you know that it's not Pseudomonas. All right, so again, other bacteria have electron transport chains that look a little bit different. For example, E. coli have slightly different looking electron transport chain. The idea is the same, right? E. coli also produce ATP by making electrons move through a series of molecules in its cytoplasmic membrane. But the molecules that it uses are a little bit different, and here's how it's different. One thing that differs is this. It turns out that the electron transport chain in E. coli does not have a complex 3. Neither does it have a cytochrome C. So it's kind of a more shortened version of an electron transport chain that you'll find in E. coli. So the electron that ends up being delivered to the start of the chain ends up going directly then from coenzyme Q to complex number 4. So there's none of this kind of stuff in the middle here. If you notice, this complex number 4 in E. coli, what it's doing is that it's grabbing an electron from coenzyme Q. So because the final complex in E. coli is oxidizing not cytochrome, but this coenzyme Q molecule, complex 4 in E. coli is not called a cytochrome oxidase. Instead, it's called ubiquinol oxidase. So coenzyme Q is kind of very similar structure to ubiquinol. So the complex 4, the last complex in E. coli, what it does is that it oxidizes or takes away electrons from a molecule called ubiquinol or coenzyme Q, not a molecule called cytochrome, because E. coli don't have cytochrome C. Right, so that's one difference in the electron transport chain of E. coli. It lacks complex 3, and it also lacks cytochrome C. Another difference that we'll point out is this. It turns out that in E. coli, they can have two different types of complex 1s, right, depending on the type of E. coli. So the difference between the two types of complex 1s is this. So some complex 1s end up pumping protons and others don't. And so depending on what is being used as the electron source, E. coli then will use a different type of complex 1. Another thing to point out in E. coli as far as how their electron transport chains can differ is this. It turns out that different E. coli can have different types of complex 4s. Not only that, but different types of complex 4s can be produced in the same E. coli bacterium, depending on the type of environment that it lives in. And so the difference between the types of complex 4s that a bacterium like E. coli can produce is basically their how well they attract oxygen. Right, so if you take a look at the function, or if you consider the function of complex 4 of the electron transport chain, not only does it take electrons from the previous molecule in the chain, but the other kind of really important job of complex number 4 is that it's responsible for bringing whatever molecule is being used as the final electron acceptor to the electron transport chain. So here we're taking a look at an aerobic respirator like E. coli. If it's an aerobic respirator, it uses oxygen as its final electron acceptor. The way that oxygen gets to the electron transport chain is that this complex 4 is basically attracting that oxygen so that it can become part of the electron transport chain. So what we're saying here is that there are different types of complex 4s that E. coli can use. And the difference between those complex 4s is their affinity for oxygen, how well they can attract oxygen. So there's one type of complex 4 that some E. coli can use that has a relatively strong affinity or a relatively strong pull for oxygen. And E. coli would use this complex 4 when it is living in an environment where oxygen is kind of lacking. So if E. coli is living in an environment where there's a really low concentration of oxygen, then it would make sense for it to use a complex 4 that's really good at pulling oxygens towards it, right? It would want to give itself a better chance of attracting what little oxygen there is in the environment. The other type of complex 4 that may be used by E. coli differs because it doesn't have quite the same pull for oxygen that this other complex 4 does. Right? So there's another type of complex 4 that has a weaker affinity or a weaker pull for oxygen. The situation where E. coli would want to use this type of complex 4 is if it lives in an environment where the oxygen concentration is really high. And so the idea behind that is this. If you're an E. coli cell and you live in an environment where there is just a crap load, there's a ton of oxygen, you kind of want to regulate how much oxygen you take in. Because if you take in too much oxygen, it turns out that can be toxic for a cell. So you might remember from cell biology that some of the byproducts of oxygen metabolism include oxygen radicals. And so if you're taking in too much oxygen, you're producing a lot of oxygen radicals that can actually end up damaging the cell. So if you live in an environment with a really high concentration of oxygen, you want to kind of regulate how much of that oxygen you actually take in. One way of regulating or limiting how much oxygen you take in is by using a complex number 4 that is not as good as attracting oxygen towards it. One other thing to mention about this type of complex 4 is that it differs from the other type of complex 4 not only by how strongly it pulls oxygen, but also by how many protons it pumps. It turns out that this particular complex 4, the one that has a weaker pull for oxygen, actually pumps more protons than the other type of complex 4 that we just mentioned. So to kind of sum this up, we're saying that aerobic respirators like Pseudomonas and E. coli, they basically use the same process here as far as electron transport chains are concerned in order to generate ATP. There can be differences, though, in the components that make up the electron transport chain. What we'll do next is look at or mention one other way that electron transport chains can differ among bacteria. And so we mentioned before that there are bacteria called anaerobic respirators. And by definition, if it's an anaerobic respirator, it's using an inorganic molecule besides oxygen as its very last electron acceptor in the electron transport chain. So if it's an anaerobic respirator that's not using oxygen as its final electron acceptor, that means that some of the parts of its electron transport chain have to be different. So here's what we mean by that. All right, so again, if it's an anaerobic respirator, it's not using oxygen. Instead, it uses other inorganic things as its final electron acceptor. And so because anaerobic respirators need to attract things besides oxygen, that means that its complex number 4 is going to have to be different as compared to the complex number 4 of aerobic respirators. So to kind of give you a visual, we'll make it a little bit different color. So the complex 4s of anaerobic respirators have to be different. And again, the reason why is so that it can attract other things that aren't oxygen. So things like nitrate, that's an example of something that an anaerobic respirator might use as its final electron acceptor. Turns out that after nitrate is reduced, it becomes nitrite. Another thing that differs in anaerobic respirators as far as its electron transport chain components is this. So remember this coenzyme Q, its job is basically to take electrons from the start of the chain to the next complex in the chain. It turns out that coenzyme Q only works with the complex 4s that are found in aerobic respirators. Coenzyme Qs don't work with anaerobic complex 4s. The reason why is because the structure of anaerobic complex 4s is a little bit different than the structure of aerobic complex 4s. So this molecule is not compatible, would not be able to bind to it. So anaerobic respirators then can't use this coenzyme Q that aerobic respirators do. Instead, they use a molecule called menaquinone. And it turns out that this is a molecule that is compatible with complex number 4s of anaerobic respirators. This guy can bring electrons to this anaerobic complex 4. So again, what we're doing here is taking a look at some of the differences that you would find in the electron transport chain of anaerobic respirators. To sum up, here are the differences. Anaerobic respirators use a different complex 4 that's capable of bringing in a different inorganic electron acceptor besides oxygen. And also, anaerobic respirators use a diffusible electron acceptor. In other words, something like menaquinone that's compatible with this complex 4 that anaerobic respirators have to use. All right, so to kind of sum up what we've just taken a look at, here's the big picture. We're taking a look at metabolism of cells. We're saying that metabolism is basically all of the chemical reactions that have to happen inside of a cell in order to keep that cell alive. In order for those chemical reactions to happen, there are basically a few molecules that a cell has to have access to in order to stay alive. Cells have to have access to an electron source. They have to have access to an electron acceptor. The reason why these two things are important is because when electrons move between them, that's what generates a whole bunch of ATP. Cells also need a carbon source in order to stay alive, because carbon sources are what's used to build the bigger things inside of a cell. We mentioned that you can categorize cells based off of what type of molecule they use as these things. And so what we've just taken a look at is this. We've just taken a look at cells that can use organic molecules as their electron source. By definition, that would make that cell, again, a chemoorganotroph. We've also just taken a look at cells that can use oxygen as its final electron acceptor. And again, by definition, that would make that cell an aerobic respirator. We've also taken a look at cells that can use organic molecules as their carbon source. By definition, this cell would be described then as a heterotroph. Right? We've just seen that something like glucose could be used as both an electron and a carbon source, right? Because it has high energy electrons in it, and it has carbons in it. Glucose happens to be organic, right? So if a cell is using glucose as both an electron source and a carbon source, then it would be described as a chemoorganotroph and a heterotroph. You can combine those terms and just call it a chemoorganoheterotroph. Right, so kind of just in a nutshell, here's what we've just taken a look at. Here's what we'll take a look at next. So we'll take a look at a different category. So next we'll take a look at cells that use inorganic molecules as its electron source and inorganic molecules as its electron acceptor. These types of cells, they make ATP using the same idea. Right? It's all about just electrons moving from the electron source to the electron acceptor. That movement of the electron represents kinetic energy that can be used to make ATP. So the overall idea is the same when you're talking about chemolithotrophs and anaerobic respirators, but there are some differences. And so what we'll do is take a look at some of those differences.